everybody. Here we are again. ISCT TV is back and coming to you from Paris. Cheers.
Cheers.
This is a reminder that we have the concept of ISCT TV, and it's supposed to be informative, engaging discussions, and showcase ISCT community. This year we have even more content, including exclusive behind the scenes. We also have interviews with the ISCT leaders and interview with our corporate partners to keep you up to date on the latest and greatest in CGT.
And I think I speak for both of us when I say that we're thrilled to be here for our third year.
Yeah,
Not-
right here.
Not just because we get the orange berets or the wine or the jackets, but because we're happy to be here, and it's really an honor to be here to summarize all the content and work closely with the ESP ambassadors. And so I'd like to introduce those ESP ambassadors. So first off, we had Ted Kiefer, we have Alicia Didsbury, and Ricardo Martin Santos. So to kick off the first ISCT TV of ISCT 2023 Paris, we will be joined by the ISCT 2023 Paris co-chairs. Coming up next.
Hello, and here we are again, and we are with the co-chairs of the ISCT 2023 meeting. So we have Julie Alexan, Phil Vanek, and Massimiliano Necchi. And I'm introducing them today, and we want to talk about the importance of the ISCT 2023 theme, and is celebrating the progress of advanced therapy, building the future through translation. And I want to know what that means to you.
Well, so we have discussed a lot about the theme of this 2023 meeting on these teleconferences, this late night for me. Typically it was between midnight and 2 o'clock.
Oh my
Not for all of us.
Not for all of you, because we had to accommodate people from US, from Asia, and from Australia. So a bit sleepy at the time, but there were 2 or 3 different proposals, but this seemed to us the most striking and the most appropriate for us because of many reasons. Well, first of all, celebrating comes from the fact that since long time we didn't get together. Well, it was last year in San Francisco, but still not really. While we were really thinking about back in 2020, the meeting should have been in Paris, but we couldn't for the COVID pandemic, and so let's celebrate. This is the reason why we decided to put the word celebrating, and Jacques was really pushy on this. Push forward the science towards the clinical translation, because we are in a period, as I said before during the introduction, where we really have everything in place to push cell and gene therapy forward and going from the bench to the clinical bed. We have seen amazing achievements in the field of cancer with the CAR T cells, of course, and we're looking for more to come from iPS cells, induced pluripotent stem cells, but also gene therapy with new vectors. And so we are really in an exciting era, and we have everything we need to really go from the bench to the bedside. So that's my feeling about this team of this year. But I would like to know also a little bit more from the co-chairs.
Yeah, you can add.
Yeah, no, I think as we're celebrating the successes. We see close to 30 products that have been approved by FDA and commercialized. So that's really exciting. When I think about it, gosh, I've been in ISCT, well, I was one of the founding members, so it's been over 30 years. So it's changed drastically. I think that's part of the excitement. The other part of the excitement is not just the cell therapy, but gene editing, genetically engineered cells. It's exciting. It's there. It's, it's in clinic. It's been commercialized. And then yes, and then we're thinking about what's next. Keep translating. We're looking at bioprinting, tissue engineering. There's a lot of excitement going on in that area, especially with D-cell, ReCell, and then hopefully bioprinting. So I think that really is the excitement. I mean, we've all been waiting for this. We've got a lot of tools in place, a lot of things that are GMP ready, and so it's exciting.
Yeah, absolutely.
Thank you.
I think 10 years, or going back 10 years, if somebody said CMC and, you know, distributed manufacturing, people would have looked at you, what does that mean? But here we are looking at 2023 and onward, and these are topics that are very du jour and very on topic. So the conversation about representing the manufacturing side of the industry and the launch and patient access program, it's really exciting to see not only celebrate the past, but to see that there's a real industrial or perhaps a just patient access thread that now sort of is helping to reinforce the good science and show that there are patients at the end of this.
Yep. So it's important to remember. All right, so in 30 seconds, I'm going to ask each of you to tell me about the highlight of the day for you.
30 seconds.
Next.
Well, to me, I have to go emotional. And to me, so the session, the presidential session, and the introduction before the presidential session were something exceptional. We really work hard to organize this meeting, and you know, being up there on the podium, trying to welcome the people at the meeting, and to try to tell them the passion that we put in this program and to build this program. It was very something nice for me, so I have to go with that.
That's perfect.
And you
Yep.
did.
Bravo.
Yeah. Good show.
Okay, that really is mine too, but I just want to say that I led a roundtable with 3 speakers today for induced pluripotent stem cells market readiness. The room was filled, they were spilling outside, it was amazing. And it's so exciting to see everybody's interest in that and what the potential is of that. So super, super excited about that.
I don't mean to take away from that, but that was almost every session. People on the floor.
Yeah.
It's amazing.
Yes, that's
Yeah.
why we do the roundtables.
Yeah, 2 things for me really briefly. The first is the fact that I got blocked out of rooms I couldn't get into because they were too full at capacity and beyond, and people, as you said, sitting on the floor. The second is recognition of the award winners. I think very well deserved. There's lifetime achievement, and the others, everybody, you know, kudos to them for just moving this industry forward, and that really kind of left a tear in my eye a little bit. So.
Oh, good.
Yeah, I completely agree. Thanks for bringing that up.
Very sentimental.
Yeah.
And then another 30 seconds. I'm going to hold you to this one. What are you looking for most for the rest of the conference?
Well, I'm really looking forward the plenary session of tomorrow. The plenary session I will chair is going to be really exciting, I believe. With people talking about it's really 3 speakers going from scientists talking about basic science, someone else talking about, and I'm not going to reveal the names, someone talking about the translation, and someone with a clinical application of a new cell therapy. So I'm really looking forward.
I could tell you the plenary session that I'm chairing, but I will tell you networking. It's amazing here. I'm so excited to see everybody. It's been, 30 years of meeting people and learning. There's a lot to learn here, so I'm super excited about networking without a mask.
That's right. I'm looking forward to my first sip of wine, but beyond that, I'm very much looking forward to not only the plenary sessions, but also the number of concurrent sessions that are really well organized and I think very topical for what we're trying to achieve.
Perfect. So I want to thank you so much, Julie, Phil, and Max, for joining us. And I want to say that next we will have a behind-the-scenes interview from TED with Bambi Grilli, ISCT Chief Regulatory Officer, to tell us about the first ever ISCT Global Regulators Summit, which took place yesterday. But I don't want to say until next without having a salute from all of us.
Salud.
Salud.
Salute.
Chin chin.
Chin chin.
Hello, Bambi. It's wonderful to have you here. I'm so excited to have you here for
Excited.
ISCT, ICT TV. How long have you been with ISCT? I'm a novice. I've only been with ISCT for, I think, 2.5 years. So
this is kind of complicated because I cannot remember my first ISCT meeting, but I think my daughter was 5 and she's 26.
okay.
So I'm in the neighborhood of 20 years with
All right.
ISCT.
Any advice for a newbie? We're ESPs. We got to learn a lot coming to these sessions. What's been important? How has ICT changed during the course of you being here?
Yeah, I think that the ways it stayed the same is that I always think that this is a very small community at heart. So when you come to an ISCT meeting, you see some of the same people, and it's like coming home, right? You're hugging people that you only met at a meeting and that you've only seen once a year at a meeting, and it still feels fabulous, like they're your old friends. I think that the most important thing is to really take advantage of that, right? And to make sure that when you come to these meetings, you don't just go to the sessions that are easy, or the sessions that are your peers, or even the sessions that are specific to your role in the field, but to try and get into different things, to try and meet different people. Because what I found is as you go through a career, you run into situations where you think, oh my gosh, I met this guy who can help me solve this problem. And you just met him because you sat with him at lunch, or you had a session with him and you talked to him. And then it kind of comes full circle and it helps you move forward with your job. So I think this is an incredible place to meet a lot of different people.
It has been fantastic for me, without a doubt. This is my second year as a ISCT ambassador, so really excited about that. And everybody knows Bambi. Obviously, as you can see, she's very engaging. She's working with a lot of people. What is ISCT doing to facilitate some of those less than potentially comfortable interactions with regulators?
Well, first of all, I think that ISCT is the only organization I'm aware of that has an international footprint in cell and gene therapy that's really trying to pull together different regulators, trying to have dialogue about what regulations exist in what parts of the world, how those could be improved, or how those could be standardized. I think we're going to talk about this a little bit more later in the summit. But to me, the best part of the summit was that different regulators were presenting their regulations, their footprint, and pointing to the weaknesses and learning from other regulators that had different weaknesses. And so it was an interesting interplay to me to say, oh, this is how regulations improve.
Yeah.
It's when regulators get together and say, oh, gosh, it's interesting you did that. Maybe I could try that, or maybe we could tweak this a little bit.
Sure.
So it's really interesting. And by the way, I do think regulations are really interesting. I think it takes a certain kind of person, but it's almost like gambling or something.
Yeah.
It's like you know the rules and you're trying to figure out the best way to play the game.
Yeah.
That's what regulations are.
It has to be operational. Regulations have to be broad enough to keep everybody safe, but they got to give you the freedom to develop your own products.
But the problem is, and I want to be clear about this, the problem is that a lot of regulators view themselves as being a roadblock. I don't consider regulatory affairs to be a roadblock. Regulatory affairs is understanding the law well enough to figure out what you can or can't do, helping investigators and physicians and sponsors do what they need to do to get products to patients. Otherwise, you're not a good regulatory affairs person.
I mean, I'm a quality control person. I'm a quality assurance guy. So I mean, that's kind of what regulatory is to me.
Right.
And it's just making sure that everybody's on the same page and managing. Yeah. So you mentioned the summit. So the summit is something that ISCT did yesterday, correct?
Yes.
You want to go into some details of exactly what that was?
First of all, I do want to say, because I forgot to say this yesterday, that Karen Nichols should get tons of kudos because she was involved in setting this up initially. If the pandemic hadn't hit, she would have been the person who did what I did yesterday.
Okay.
And unfortunately, she wasn't able to. So all hail Karen Nichols, who should be here getting praise for this. But she wasn't, and I am. So it was a lot of fun. We had 14 agencies in the room, more regulators. We had some agencies who sent one person. We had ISCT leadership. We had everybody from the ISCT Global Regulatory Task Force in the room. And we spent the day talking about unlicensed, unapproved cell therapies or stem cell therapies. and the proliferation of what we call bad actors and clinics that are not properly providing such products to patients. We talked about, and that was where it got interesting to talk about which agencies were approaching these things in which ways, and what was working and what wasn't. And I think that, I talked about the fact that there was some gaps where you could see people literally thinking, oh, that's interesting.
Yeah.
I could learn from this. But you could also see a lot of similarities. And I thought one of the most interesting similarities was the discussion of the fact that in regulatory affairs, when you get into real bad actors and you get into people who are breaking the law, hurting patients, we don't have a strong punishment process for that gets outside of the criminal law.
Okay.
And so, there's a case in the U.S. right now that's gone sort of this way, right? Where the regulatory part, I think most of us feel clear about, but this is playing out in a standard court of law with a judge who's not a physician and be a PhD. And so what are his decisions? Well, they're not going to be the same decisions we would make because he doesn't have the same understanding of what we're talking about on the table. So it's interesting to see that that's actually one of the weaknesses of all of this, is that when you talk about these clinics and you talk about, inappropriate stem cell usage, you're talking about only being able to pursue that in a regulatory environment that doesn't have like an associated jail and things like that. So that was very interesting. So that was a commonality across all of them. I think everybody in the room said
that's
the same.
That's a very unique perspective. I mean, I hadn't been thinking about that until this point.
The truth, and I'm 20 some odd years into this, so it's very interesting to actually think about, oh, that's why these things can happen, and then you can see where the bad players, the bad actors, they're looking at this and there's like, here's the benefit. I can charge, I don't know, let's say $200,000. I can charge this patient $200,000 to give them use therapy, and I can maybe get away with it 50 times. And
that's a big price tag.
And I have to pay $50,000.
Yeah.
Or whatever. It's worth it. It is worth it, right?
Yeah, the cost risk is.
Yeah, the risk benefit there is not in favor of following the regulations, and so that was also very interesting. So
yeah, that is very interesting.
It was a lot of fun. We really had a good time, and there was an enormous amount of dialogue about a lot of things in the room.
I love it.
So.
Sorry, I didn't mean to cut you off, but I love those sessions with an enormous amount of dialogue.
Yeah,
and we've had a couple today.
Engaged. Everybody was engaged, which was also nice. I didn't feel like any regulators were just like, I'm not learning anything. I hate this.
I'm assuming you had FDA there.
Yes.
EU. And like ISCT also, approaches this as a global solution. So it's not just the big payers, or not the big regulatory bodies that are going to be involved in this. Some of the smaller ones.
I don't want to go through them because I'm not sure I'll capture everyone, but we had someone from Israel. She's sitting right there. We had somebody from Japan. We had Korea. We had Hong Kong. Canada, the UK, and then we had with the EU, it's a semi-combined and semi-disjointed, right? So there was also Austria, there's also France. So we had other players at the table. So it was interesting.
Kind of like state regulatory bodies in the US. Yes. You have the FDA, but you also have state.
Yes. Although it plays out differently in the US, right? So it's anyway, it was very interesting. So we had the European Commission, and we also had the EMA. So that was also interesting to see, because they had an actual interplay at the table that was interesting to watch too. And I think that's part of this is, if anybody had the perfect answer, we wouldn't be having these meetings, right? But we don't. So we're still trying to figure our way through. Australia was there. I do not want to forget Australia for sure. That would be bad.
Well, anybody who was there, I'm sure, will give you forgiveness for not being mentioned. Not a big deal. So, this isn't a plug. This is just opportunities to say that ISCT is bringing together as many people as possible, extending invitations, making a global platform.
It was fabulous. And, everybody in the room said the same thing. They'd never seen that volume of high-level regulators. These were not just some guy who worked at the FDA for a year, right? These were high-level regulators in the room talking about serious issues, and it was really fun. And we're going to do it again next year. Can't wait.
Fantastic.
So I think that
we're looking forward to it.
Yeah, it'll be fun.
Yeah, and so all those regulators who came in were also invited into roundtable sessions, or a lot of them were invited into roundtable sessions, I believe, today. Yeah, well, I mean, once you're here, let's take full advantage of the opportunity.
Thing. Listen, you're doing something else
Yeah.
too. So yeah,
Well, let's share that knowledge. Let's get it out.
I think most of them were at roundtables, and in fact, quite a few were at the Global Regulatory Task Force at a roundtable that we had this morning,
Mhm.
which was an interesting. We had a case study, a fairly complicated case study. Our first session was really about CMC issues. Our second session was about preclinical,
Mhm.
and then our third session went into potency.
Yeah.
And all of them were interesting. All of them were highly attended. The room was standing room only, and we didn't have a lot of people leaving. We just had a lot of people coming in. So that's always good when you're at the front of the room and you see
Yeah.
people coming in, not fleeing out the back. But the one that I thought was the most interesting was not. I was preclinical. The potency one, because potency is something that we talk about it, we talk about it, we talk about it, we keep circling it. People got really heated in there about potency. And in fact, one person got up to ask a question and said, we've been talking about this for 10 years. How many more years are we going to talk about this? That's pretty heated for
That
a
is.
roundtable.
Yeah,
right?
for scientific discussion.
So it was fun.
It was fantastic. And you
Yeah.
did a wonderful job moderating the preclinical. I was there for the, of course. Charming
Enjoyed it.
is all get out.
Well, the group was so fun, right? I
Yeah,
had
it
a
was
great group.
fantastic. Well, I think that's about all the time that we have.
I know.
So this
Talk
has
your
been-
ear off.
Oh, talk my ear off all the time, please. Anytime you get an opportunity, I'm happy to hear what you have to say. So thank you so much for spending time with me.
Enjoyed it.
Yeah, we'll have to do this again sometime. Maybe next
year. We'll look
All right.
forward to it. Thank you.
Au revoir.
All right, and we are joined by one of our ESP ambassadors, Alicia Didsbury, who has joined us all the way from the land of the Kiwi in New Zealand.
It was a 38-hour journey, but it's so wonderful to be here.
Alicia, tell us, what have been your highlights from day number one?
I have so many highlights. It's been an amazing day, and this is actually my first ISCT global meeting, so it's been quite intense. And I started off very early at the first roundtable session, and actually I attended both of the EU regulatory sessions focusing on hospital exemptions, and it was really interesting, especially from my perspective in New Zealand, where we don't have any regulatory issues to speak of because we don't have anything to regulate currently. So it was incredibly informative. Some of the key concerns, they were talking about balancing patient access to therapies with providing safe, effective therapies, as well as having those therapies go through the appropriate regulatory pathways. So it was very enlightening conversation for me.
And I think those same concepts I've seen come up in multiple other sessions throughout the day. I think it's something that's right at
The forefront,
Yeah, exactly.
And it's been really interesting, the contrast for me between the European Union and then America, and then for us at ANZ, and the different spaces that we're in, and how to deal with those challenges that we have, which are actually all unique to our regions. And then one of the big highlights for me was the ESP roundtable session. So this was the COVID hangover, how do we restore public trust? Mm-hmm. And again, for me, this speaks very much to the things that are close to my heart. I'm all about communicating what we do to the public, and in a way that's meaningful and genuine. And so one of the big questions was, what's our role? What role do we play in ensuring that the public can make informed decisions on the products that we're making? How do we navigate the sort of snake oil sellers, so to speak, and how do we, at an institutional level, deal with some of the patient questions where people are genuinely desperate for treatment and they really just want to be offered hope. So it was actually quite a moving session, and incredibly well chaired. It was amazing.
I find that we do have to preface every conversation about those regulatory considerations with it needs to be appropriately regulated, but there might be a different way to do this.
Exactly, yeah. So I mean, it's obviously an ongoing conversation, which is incredibly fruitful. And then speaking of ESPs, I would really love to introduce our ESP co-chair of the mentoring work group. So we had a mentoring work group session today, and Nisha has come to speak to us about it briefly.
All right, hey everybody, it's my pleasure to introduce Dr. Nisha Duran. Did I say it right?
Yes. Got it right.
Can you summarize the ESP session that happened earlier in the mentoring session?
Sure. So earlier today at 3 p.m. in this very room, we had an ESP reception, mentoring reception. So basically, throughout the year from November to around June or July, we run our flagship mentorship program, and we decided to host this event to give an opportunity for mentors to meet with mentees beyond the people that they are paired with. So that is what the event was all about, to encourage networking and relationship building. And also we had a very good panel discussion with 3 elite panelists. In the past few years, we have really had a good turnout from mentors, and that has allowed us to have 1-to-2, 1-to-3, and one-on-one pairings.
If I can interrupt you 1 second,
Mm-hmm.
how many mentees are in the program now?
Oh, it's over 100. So this year, the total number of participants, both mentor and mentees, is 232.
Wow.
It has grown tremendously. That is the most number of participants we have had in the program to date. And hopefully in the next iteration of the program, we would like to see it grow even further.
That's amazing, especially considering the importance that ISD places on mentorship and workforce development. I think that it's great to see that there's been such uptake of that mentoring program.
Yeah, so I agree. It's just been incredible growth that we're seeing in the program throughout the last couple years. So back to your question about once we do those pairings, we have a kick-up call in November where we just give some background about the mentoring program, introduce participants to each other, and then basically from that point, it is up to the group, the mentor and the mentees, to decide when they want to meet. We encourage them to meet once per month for the next 8 months. And then in addition to these one-on-one mentoring meetings or sessions, if you want to call it that, the mentoring working group also puts on other activities throughout the year. So this year we had a recruiter workshop where we got recruiters from industry and academia to give tips on writing the resume, updating your LinkedIn profile and CVs. And then we also had small group meetings. So beyond the core activities of the monthly meeting, we try to encourage or we try to develop other programs for which participants can be involved with.
I just want to say that if you're not part of this group, you are missing a great opportunity. It's amazing. I've been a mentor for many years and I enjoy it immensely. So please, if you haven't participated, just contact us, contact ISCT, and find how to be part of it, because you can say something. Yes, it's amazing. Well, that's all I can say. Yeah.
Yes, I agree. It's a very good program. I've been a participant since I have been involved with ISCT, and we have experienced tremendous growth over the years, and I'm just looking excited to see the program grow even further and expand our reach even further.
Yeah, and just touching on what we had talked about earlier, in addition to the mentoring program, I know that the IST has created a workforce committee.
Mhm.
They have the CMAT workforce manufacturing programs. I know there's 2 other programs in Europe. So just seeing that portfolio grow is amazing.
Yes, I agree.
Bonjour, comment ça va? I'm here with Bernd Giebe of Université Hospital Essen. That took a lot of practice, so I'm glad that I got it out right the first time. I'm Ted Kiefer with ISCT TV, and we're going to talk about exomes and MSCs. So you have been prolific, or not, I mean semi-prolific, prolific in publications, especially here recently. Do you want to talk about some of your current publications and your thoughts as to why the MSc community is revitalizing, or at least I feel like it's revitalizing, because there's been a lot of thought, there's been a lot of talk about MSCs not necessarily being the focus of advanced therapies.
Yeah, so at first, thank you for the interview. And so I think we're all aware that MSCs have enormous therapeutic potential, but it's also discussed controversial. Some works, others do not work. And so we thought, can we overcome it by maybe taking the active component of them, which are the extracellular vesicles, small extracellular vesicles, which in the industry are termed exosomes. We would say exosomes are just from the endosomal system. But indeed, we showed in animal models that these EV preparations, as we would say, are very therapeutically active. And conventionally, we just publish positive results, right? So, so if you apply them, they have great effects, but we observe not all preparations have these active effects. From time to time, there is a preparation which has no activity, and we have some MSC stocks where the EV preparations hardly have any activity. And that is what we elaborated and demonstrated on the one hand in a graft-versus-host disease mouse model, that there are potent and non-potent preparations. We established then an in vitro assay, which scored the activities according to the GVHD, which we call the multi-donor mixed lymphocyte reaction assay. And finally, the heterogeneity and the variability in the efficacy. We think it's due to polyclonality of the MSCs, that there are different relationships, that one population becomes dominated in the one preparation, the other in the other preparation, so that there is variability, stochastic variability. And to overcome it, we thought, can we now immortalize the MSCs, which we did with a lentiviral TERT-based method. And then we selected single cell clones. So we deposited single cells, expanded them, and now have monoclonal cell lines. And the question was whether these EVs retain their activity, and they did. That is what we published in a hypoxia-induced encephalopathy mouse model together with Josefina Herz.
And I'm very excited to see where this goes from a clinical perspective, because individuals are very different.
Yes.
We have a heterogeneous recipient population. Homogenizing the donor population or the cell line is going to make, I think it's going to blow some heads, in all honesty.
Yeah, that is our hope, that we can, on the one hand, scale the product much more. And since we have a larger batches of the drug, that we can treat more patients, and then also figure out how many people are really responders and how many are not responders, and maybe then also find criteria to exclude the non-responders right from the beginning from clinical studies. Because if you have too many non-responders,
it can kill a therapy.
It can kill the effect in phase 3 clinical trials.
Definitely. Yeah. And this ISCT is all about getting as much therapy to patients as quickly as possible. So I'm very excited to hear that they've engaged with the exome community, the MSC community, and they're not only inviting you to moderate roundtables. They have another session planned for Saturday. Can you go into the details of that and the goals, why it came about?
Yes, so I think, so I'm heading the exosome committee together with Sai-Keng Lim, who is the pioneer for me in the therapeutic EV field. And when we read on average reviews and we discuss how do EVs work, then it's quite clear. So they the vesicles contain RNA, microRNA, and they are uptaken by endocytosis, and then they deliver the luminal cargo, and this is the active component. And this, to our latest understanding, is not true. So apparently there is a big hurdle to deliver luminal cargo into cells. You need fusogenic proteins, or you need to have endosomal escape machineries, which normally are not presented. And I think we need to educate the society about the principles of the basic EV research. And so we thought it's a good idea to have a special session about exosomes. So, and Cycling and I managed to get some leading experts. So Clotilde Terry here from Paris. For me, it's the grande dame of exosome research, and she's one author of the MySafe criteria. And we have Guillaume Fanil as a speaker. He worked with zebrafish and can track yeast in a living organism. Very exciting data. Yeah, and so we have more speakers, and I think we hope that we can convince the community that maybe the EV biology is a little bit different than many think, and bring them to the current state of the art.
Go figure that cell and gene therapy is a little bit different. And mesenchymal stem cells and EVs being quoted black magic of advanced therapies means that it's that much further removed. I know that's not a great term, but you mentioned controversial. The thing that eliminates controversy is setting a baseline and getting everybody on the same page so that they can understand the next level.
I think in science we are driven by dogmata all over, and so we know it from the time of Galileo, where the Earth was a disc, and then he and others claimed it's a ball, and that was dangerous. And I think we still have today many of these dogmata in our scientific world. And yeah, maybe this educational also helps to get some of these dogmata out of the mind. Maybe we just see the new dogmata and also are wrong. Yeah. We are limited with our technical abilities. So until recently it was everything which cannot be seen in the light microscope, which is not infectious, was studied as a homogenate.
Yeah.
And with that, we oversee the nanocosmos in our body liquids. And I think there is a lot of things in there. They are not just EVs, there are other particles in the 100 nanometer range. And now it's a challenge to get techniques where we can start to dissect this 100 nanometer cosmos and work with it.
Microfluidics and the dynamics of microfluidics sound fantastic. People that I need to introduce you to, you'd have a great time. I love the fact that you can marry philosophy and science and understand, the influences outside of that. So it's been wonderful speaking with you. I'm going to let you get back to the plenary sessions, which are going to be fantastic.
Yes.
And I hope our paths cross with some beer or some wine moving forward.
Yes.
Beaucoup de vin.
So thank you very much, Ted. It was a pleasure being here, and so enjoy the conference. Thank you very much.
Take care.
All right. Well, as a wrap up, I just want to say it was a real pleasure to speak with Phil, Julie, and Max about the inspiration behind the theme for ISCT 2023 in Paris. I think I'm really looking forward to tomorrow with the networking events, with the industry networking, and of course the ESB networking. I think that's going to be fabulous. What about you?
Oh, I love to talk with Alicia and Nisha, the ESB co-chairs. I love being a mentor and having my mentees, and I love what they're doing with the whole thing and how many new mentees and mentors we have. So that was amazing. I'm looking forward to the networking, and I'm also looking forward to, I'm gonna do a round table on Saturday. I'm a moderator about ISBT 128 and the new split label that can help the industry. So that's another thing that I'm looking forward. Of course, the plenaries that are always so informative, and they're great. So I'm excited. The first day went amazing. I want to go out there to talk to people, because I haven't had really time to talk to many people. Because it being a day going to all the sessions, and I have a great time. So,
well, I'm happy to be here with you again.
Me too.
Yep. Cheers.
Cheers.